Hello, my name is John Moyer, and I want to welcome you to the NIH Office of Disease Prevention's Mind the Gap webinar series. This series explores research design, measurement, intervention, data analysis, and other methods of interest to prevention science. Our goal is to engage the prevention research community in thought-provoking discussions to promote the use of the best available methods and to support the development of better methods. Before we begin, I have some housekeeping items. You can submit questions during the webinar via the chat pod. You'll need to open the pod by hovering your cursor at the bottom of the Zoom window and then clicking on the chat icon. Please direct your questions to hosts and panelists. We will open the floor to questions that have been submitted via the Zoom chat pod at the conclusion of today's talk. The slides and video recording will be posted on our website, prevention.nih.gov/mindthegap, in approximately 2 weeks. Lastly, we would appreciate your feedback about today's webinar. Upon closing, you will be asked to complete an evaluation. We would appreciate your feedback as it will help us improve this webinar series. At this time, I'd like to introduce Dr. David M. Murray, Associate Director for Prevention and Director of the Office of Disease Prevention.
Thank you, John. Today's speakers are Dr. Ty Ridenour and Dr. Stephen Tueller. Dr. Ridenour is a Senior Developmental Behavioral Epidemiologist in the Center on Social Determinants, Risk Behaviors, and Prevention Science at RTI International. Dr. Ridenour has collaborated on idiographic clinical trials research for over 15 years in a range of fields, including prevention science, endocrinology, intimate partner violence, psychotherapy, physical rehab, and wearable biosensors. He has also taught graduate courses on idiographic clinical trials and leads development on the PersonAlytics R Package, funded by the National Center for Advancing Translational Sciences. His content areas of interest are in the etiology and prevention of substance use disorders, unsafe sexual behaviors, and conduct problems. Dr. Tueller is a senior research statistician in the Division of Applied Justice Research at RTI. Dr. Tueller provides design and analysis leadership for several ongoing multi-site randomized trials, hybrid trials, idiographic clinical trials, and implementation studies in research areas spanning child welfare, juvenile justice, human trafficking, rare diseases, HIV services, adult criminal justice, and addiction. Current projects are funded by the Administration for Children and Families, the NIH, the National Institutes of Justice, and Arnold Ventures. Dr. Tueller's current methodological work focuses on developing statistical models for medication adherence research. He also leads development of the PersonAlyticsPower R Package. We look forward very much to this presentation, and I will turn this over to our two speakers.
Thank you much, David, and thank you all for taking some of your valuable time and attention to attend this presentation. Stephen and I would like to start by acknowledging that funding from NIH's National Center for Advancing Translational Sciences is supporting this presentation, the power analysis simulation research, and the software that we'll be describing. More detailed reports on each of the studies that I'll be describing and others are available at the website that will be shown at the end of the talk, or you can get in touch with either of us for those. That the website that I referred to appears at the bottom of this page. Use of idiographic clinical trials, or ICTs, has expanded over the last decade, but is still not widely used at this time. So we'll start by describing some of the scenarios when ICTs can be especially useful for addressing gaps in the more traditionally used clinical trial designs. Next, I'll describe the fundamental building blocks of ICTs and note some of the more common options that are used. I've found that examples of idiographic clinical trials are worth thousands of words in terms of grasping the nature of the evidence that comes from them and the advantages of these studies. So I will review some of the more recent methodological developments in this rapidly evolving methodology after showing examples of ICT studies. Stephen Tueller will then describe in depth the PersonAlytics software programs, including how to conduct power analyses for planning ICTs. Many of the advantages offered by ICTs are listed here. One of the most important advantages is they can provide rigorous methods for experimental studies that are limited to small samples. By small, I mean fewer than 100 participants. These situations are surprisingly common, including treatment studies for rare diseases or pilot studies of innovative treatments. George Howe describes genetic microtrials for testing preventive interventions, and this could be an area in which these small sample rigorous designs could be especially useful. Another strength of ICTs is when a treatment study must be conducted in the wild, meaning in clinical settings when a meticulous protocol that is typically needed for randomized control trials cannot be followed, or when the external validity of a clinical trial is more important than its internal validity. Other forms of in-the-field research, when these methods can be exceptional tools, include ecological momentary assessment studies or conducting program evaluations in small settings, such as small schools or clinics in rural settings. There are many ICT study designs to provide treatment for all study participants that could be used when it is unethical to withhold treatment from any research participant, and some of these will be illustrated soon. The intensive evaluation of within-person change that is part and parcel of ICTs can be useful for identifying active ingredients of multi-component interventions, or identify the best order for delivering components of a curriculum or treatment. ICTs yield detailed evidence about within-person processes that occur during and after treatment is received, which provide detailed evidence regarding heterogeneity in treatment responses. ICT evidence is also adept for evaluating mechanisms that lead to treatment outcomes and can be ideal for comparative effectiveness research. Most ICT data collection timelines are shorter than a few months, making them useful for rapid program evaluations. Finally, ICTs can be ideal in many scenarios for testing or developing programs for underserved populations. For example, ICT use of small samples allows them to be used for initial testing of homegrown programs to collect outcomes data that may support funding of larger studies. Such programs are often developed by people with lived experiences, and examples include Big Brothers, Big Sisters, Alcoholics Anonymous, or the Good Behavior Game, each of which are among the strongest programs in prevention or addiction recovery. ICTs can facilitate evaluation of prevention programs or treatment in American Indian and Alaskan Native tribes whose cultural values are antithetical to comparing between people, because many ICT designs evaluate changes within people rather than requiring randomization into groups or comparisons between persons. This slide lists some research instances when ICTs are especially useful, and other research instances when they are not ideal. Many of the items on that first list are repetitive with the last slide and don't bear repeating. That said, the fourth item deserves mentioning because the individual participants' responses to a novel program or treatment can be reported back to them. Many people agree to participate in treatment studies because they hope the novel treatment will benefit them. So learning about their own individual response to a novel treatment can be a powerful incentive to enroll in and to complete a study protocol. The second list includes common features of traditional clinical trial studies that ICTs generally cannot incorporate, or when they offer limited utility. ICT studies have been used with very large samples, but this is very much the exception rather than the rule. And they're generally not considered for large population efficacy estimates. They're not terribly useful for acute illnesses, such as a headache that lasts only a few moments or an hour. These designs typically have timelines from days to weeks to months. They're not helpful for evaluating change in characteristics that change slowly over time or are generally resistant to treatment. These are better for evaluating and detecting change in shorter periods of time that are expected to respond quickly to a treatment. They're not useful for few waves of studies. So traditional randomized clinical trials that have baseline 6-month and 12-month or something like that are best used with the traditional ways of analyzing them as opposed to what we would be presenting now. They're not useful for surveys and large prevalence estimates. Long interviews and questionnaires, except for at baseline, are generally not used in these types of designs because they would require too much burden on the participant. And then something to keep in mind that note at the end is that for some methodologists, there's a stigma about these designs that stem from some of the historical uses, such as relying only on visual inspection of data or some of the other biases that can occur in how the data are collected and interpreted. So what composes an ICT? There are three fundamental components that are used to design an idiographic clinical trial. The first is use of a within subject experimental design in which each participant experiences two or more study conditions called phases. Many such designs exist and are also referred to as quasi-experimental designs or single case designs. N of 1 designs is another moniker for these designs used in the medical fields. Some examples include multiple baseline designs or reversal designs such as crossover studies or so-called AB-AB designs, in which participants toggle between exposure to control and treatment phases. One advantage of reversal designs is they can be varied among participants in their phase assignments to attain balance across study phases and increase the rigor in interpreting the data results. The second component of an ICT is that time series data are collected from each participant during each of the study phases. Such data may be self-reports, such as in ecological momentary assessments, but also could be acquired from student or patient records or using biosensors. These first two components have historically been used to conduct quasi-experimental designs or the other names of these same studies. An ICT differs from these studies by adding rigorous statistical analysis to evaluate outcomes and or evaluate treatment mechanism processes. I'm going to skip these two slides in the interest of time, but may come back to them later if they might be helpful for answering questions. To provide some depth and breadth to introducing the strengths of ICTs, I'll use some example studies next. This first illustration demonstrates how a multiple baseline design and intensive hierarchical modeling can be used to carefully evaluate individual differences in response to a treatment, as well as aggregate the treatment effects to quantify average outcomes in those who are treated. It also illustrates how useful ICTs can be for precision medicine research. These data are specifically from a study that was led by Dr. Thomas Pinio about 15 years ago. And while serving patients in nursing homes, Dr. Pinio observed that his residents who had diabetes often experienced uncontrolled glucose levels soon after coming into their nursing home, and he was hearing similar instances from other physicians. At that time, an antiquated approach for monitoring blood glucose was used, which only adjusted insulin doses every 2 weeks. It was referred to a sliding scale for those who may recall it or have heard of it. So he designed an algorithm and an Excel program for his nurses to enter a resident's non-fasting glucose level at mealtime, as well as the foods that the resident was expected to consume during the meal. His formula would then compute the needed bolus dose of insulin to give the resident at mealtime to metabolize the anticipated sugars. He called this approach the manual pancreas, and this general technique is widely used now in the form of glucose monitors and other treatments. Presented on the left side figures are the results for 4 patients over a period of 100 days with 3 meals and an evening snack time per day. A multiple baseline design was used in which the length of care as usual phase was staggered among participants. This staggering of the baseline periods was especially important to control for potential historical events, such as the nursing home changing their menu, which could impact glucose levels in a way that was not accounted for by the analysis of treatment. Therefore, thereafter, patients were treated using the manual pancreas. If you ignore the black trend lines momentarily to focus only on the red glucose readings, a strength of the multiple baseline approach is that sizable benefits from a treatment can be seen with the traditional evaluation strategy for within subject trials, relying on visual inspection of data, as with patient B in the upper right of the 4 individual data streams. However, as shown for patients A and C, it can often be difficult to determine if, and especially to what extent, a treatment provides benefit for research participants. By using intensive hierarchical regression modeling, those with trajectories shown as the black lines superimposed over the red glucose readings, we can quantify the immediate benefit of the manual pancreas in terms of the change in intercepts from baseline to treatment, as well as the gradual change during each phase in terms of the slope over time. By centering time at 0 for each participant, their observations and hierarchical models can be aggregated as shown in the figure to the right. As mentioned earlier, a strength of this approach is that we can provide every participant in the study with his or her individual response to the experimental treatment. And using plots like these, along with numerical outcomes, provides user-friendly evidence that can be easily understood by non-statisticians. This study is also one example of how comparisons among people is not required to estimate the impacts of a treatment or prevention program using an ICT. One note is that for using hierarchical regression model with small samples and intensive time series data, some adjustments are required. First, the best fitting error covariance structure needs to be determined to parse out autocorrelation to be able to meet the assumption that error variances are uncorrelated among model terms. Usually, hierarchical regression models assume autoregression 1, but there are dozens of potential error covariance patterns that could occur in the data, and failing to control for the correct autoregression pattern can lead to biased estimates for both random and fixed effects. A second adjustment is to use maximum likelihood estimation and common fit statistics to test whether competing predictors, treatment arms, and error covariance structures improve the fit of a model to observed data. The model parameters are then obtained using restricted maximum likelihood estimation. This is because the full maximum likelihood underestimates parameter variance components due to how degrees of freedom are allocated, and is particularly problematic for small samples. The third adjustment is that statistical correction for small sample size helps provide conservative statistical testing. One option is the Kenward-Roger adjusted F-test to reduce risk of type 1 error. Another option is the Staterthwaite calculation of degrees of freedom. And a third option may be the finite population correction to p-values and standard errors, depending on the nature of the study. Finally, in our own work, we use dummy coding to more effectively parse the effects among phases so that the output can more closely resemble a piecewise regression and be interpreted specifically per phase. This next study demonstrates a sophisticated within-subject study design for which intensive hierarchical regression was also used for data analysis. This pilot study was led by Dr. Bethany Rafe to test 2 types of contingency management schedules to increase the self-testing of blood glucose levels in adolescents with type 1 diabetes. This is because most adolescents don't come close to taking enough tests per day. The clinical goal for this behavior is for people to complete 4 non-fasting glucose tests per day, which in turn can help insulin administration and prevent diabetes medical afflictions, or at least push them out much further in time. In Dr. Rafe's study, participants were randomized into one of 2 contingency management schedules to incentivize their use of non-fasting glucose tests. There was also some suspicion that older teens and younger teens may respond differently to the treatment, and so we tested for differences between these age groups. To further complicate the study, an additional challenge was that motivational interviewing had already been demonstrated to have efficacy for increasing their number of daily blood glucose tests, and this effect needed to be accounted for. So participants were first provided with motivational interviewing prior to receiving contingency management. The study began with a baseline period of no intervention, shown here as the pre-MI phase. And on this outcome figure, you can see that during days -10 to -6, the average number of participants' glucose tests per day was 2, as shown on the y-axis. This did not change over time during this phase, with the time metric of days shown on the x-axis. Then motivational interviewing was provided to participants, and this point in time was symbolized by the first vertical line connotating the end of the first phase and beginning of the second phase of the study. Motivational interviewing had an immediate impact of increasing the average daily glucose test by almost 1 test per day on average, and that effect held steady from day -5 to day 0 of the study. I'll note the reason that we have day 0 at this point is that by positioning day 0 and centering time right before the experimental intervention begins, the statistical output is much clearer for interpretation and understanding of the treatment effects. During the first and second phases, participant trajectories did not differ by age. However, the two schedules of contingency management impacted older and younger adolescents differently, resulting in unique outcome trajectories for 4 subsets of participants as shown here. In sum, the older adolescents reached the clinical goal of 4 tests per day, with only a slight difference between the two schedules of contingency management. In contrast, younger adolescents responded better to one schedule of contingency management, although both groups of younger adolescents lost the treatment benefit after their initial exposure. After the third vertical dotted line, contingency management was withdrawn, and the trend lines illustrate that little erosion of treatment effects occurred thereafter. The equation below the figure shows the same results, but in the form of the intensive hierarchical regression equation. One implication of this study for using ICTs to evaluate treatment specifically for traditionally underrepresented groups is that rigorous pilot testing can be conducted with small samples. For example, the final sample size for this study was 41. If minority groups need to be compared to a majority group to understand differential responses to a treatment, it can be done so in a careful and detailed way without the expense and effort of a large sample study. Also, so-called homegrown interventions, like what I referred to earlier, are often developed by people with lived experience and can be pilot studied in this manner to collect preliminary data to support federal grant proposals and prior to committing large investments that are required for traditional randomized clinical trials. Second analytic approach for ICT data that I found highly informative for understanding within-person processes in response to treatment or prevention exposure is called unified structural equations modeling. The statistical model for this approach comes from linear algebra, and I'll not spend a lot of time on the statistical model because of our time constraints, but I can provide references that fully delineate this model if anyone would like. Also, the GIME program on which this notation is based, as the acronym is shown there below, it's evolved quite a bit since these citations. Unified structural equations modeling can be conducted as a series of structural equations models in which observations are clustered within individuals. Using that latter approach, the structural equations model parameters are estimated separately for each participant. To conceptually demonstrate the USEM approach or Unified Structural Equations Model approach, please consider this path diagram. This illustration comes from a pilot study led by Andrea Wittenborn that tested whether emotion-focused therapy for heterosexual couples could concurrently improve relationship satisfaction and reduce major depressive symptoms. This strategy was in part based on prior nomothetic studies or population-oriented studies that demonstrated strong bidirectional association between major depression and relationship distress. The study investigated the degree to which improved relationship satisfaction led to reduced depressive symptoms and vice versa, with each lagged pathway of influence hypothesized to be significant. About 30 couples in long-term relationships in which at least one partner experienced depression were randomized to receive 15 one-hour sessions of either emotion-focused therapy or usual care, and outcome measures were completed after each therapy session. This model also allowed us to specifically test for Granger causality. Granger causality is more about the findings of a study than it is about a methodology. And in this case, we would have found Granger causality from relationship satisfaction to impacting depression. if the pathway of S to D2 was significant and the pathway from D to S2 was not. As a quick note, these are the average trends and outcomes for relationship satisfaction and depression in men in the study over the 15-week study period. However, these average outcomes poorly summarize participants' responses to treatment in comparison to the USEM evaluation, which I'll describe next. Our USEM model was first analyzed such that all of the parameters for all participants were constrained to be equal. And these results are shown for the male participants. The fit statistics for that analysis were then recorded in that first row of the table at the bottom. Next, the model parameters were fixed to be equal for the separate treatment arms. In other words, the model was estimated separately for those randomized to emotion-focused therapy and those for usual care. This analysis did not statistically improve fit to the data. So we next estimated model parameters separately for each male participant and used their parameters in a cluster analysis to search for subtypes of responses to couples therapy. 4 clusters were identified, and when participants' parameters were constrained to be equal within those 4 clusters, the fit to the data was improved statistically to a reliable level. This slide presents the parameter estimates for each of those 4 clusters. And to summarize, one cluster's changes in depression preceded and led to changes in relationship satisfaction in a manner that was consistent with Granger causality. Another cluster's changes in relationship satisfaction preceded and led to changes in depression, also consistent with Granger causality. Men in the other 2 clusters experienced changes in depression and relationship satisfaction in parallel, but with minimal cross-lagged influences. The results have implications for potentially improving marital therapy by tailoring the therapy specifically for each of the 4 subtypes. As mentioned earlier, methodological developments are rapidly evolving to enhance the rigor and use of ICTs. One of the recent developments by Daza and colleagues is to understand ICT outcomes in terms of factuals and counterfactuals, thereby drawing close parallels to randomized clinical trial modeling. There's also been multiple simulations to inform study design. As listed here, these are some of the examples, and the last one, led by Stephen Tuller, will be described shortly in greater detail. Understanding patient preferences for study designs is a focus of a group out of Columbia University that's led by Karina Davidson. She's also, it's not listed here, but she and her group are also doing excellent work to implement analytic techniques and other methods to enhance treatment approaches that take advantage of the N of 1 methodologies and analyses. If you would like to observe some of that, get better information on it, Karina presented earlier forms of her work in a recent webinar that can be viewed at the Mind the Gap website. There also are numerous alternative designs and analytic strategies that can be cherry-picked to best align with a particular study or research question of interest. And then there's a special issue in evaluation in the health professions that's dedicated to causal mediation analysis using these types of data. Listed here are some resources that can be used with ICTs to help enhance presentation of the results and understanding of those results. At this point, I'll turn the mic over to Stephen Tuller to describe our own work to develop software specifically to support ICT research.
Thank you, Ty. We're building an R package called PersonAlytics. It builds on existing R packages that implement mixed effects models, or some disciplines call them hierarchical linear models or latent growth curve models. We use those to build additional tools to make the decision-making and some of the more tedious processes that Ty described earlier in this presentation a little more automated and a little more accessible to non-researchers. We want to be able to support analyses. We're working on building graphical user or GUI interfaces that make it even simpler to use for those who don't code in R. And Ty has been describing several methodological developments that we're trying to implement in this R package. Next slide. This is an overview of some of the things that the R package automates. We won't get into a lot of detail today. The primary goal is to give you an overview of what's available and what we're working on. So the PersonAlytics package focuses on implementing mixed effects models or time series models for N of 1 or small n intensive longitudinal designs, idiographic clinical trials, or an uninterrupted time series. There's a link to where you can download the code. For a single subject, this approach implements linear ARMA models, and for small n data and more than one, we use mixed effects models under the multi-level modeling, hierarchical linear modeling, or latent growth curve modeling frameworks. The software implements several different distributions for location, scale, and shape. There's over 70 different distributions that we'll eventually implement, and we're currently tested linear models, logistic models, and have done some work with beta regression models. The structure for time can be polynomial, time squared, time cubed. Ty's also talked about how you can implement a piecewise growth model that allows for an easier interpretation. We're building tools to automate that. And you can do several different data visualizations, which we'll illustrate in the next slide. And as Ty mentioned, some of the most valuable ways this can be implemented is with rare diseases or other small finite populations, and you can do a finite population correction to your estimates. Next slide. This is an example of the visualization from a hypothetical trial with 7 different phases and shows a distribution of the data across the different phases of the study and how things could be visualized as they change over time. And this is similar to earlier slides where we saw some real data. Let's go to the next slide. And I'll go into a little more detail into the framework. As Ty showed you earlier, hierarchical linear model has an outcome that's decomposed into fixed effects, random effects, and error. And for the distribution of the outcome or the distribution of the error, we can have normal, beta, negative binomial distributions. Those are just some examples of the 70 different distributions that can be implemented building on the GAML's R package. The fixed effects could be any of the examples Ty gave earlier, where you may have an experimental phase and a baseline phase, like in an ABA study. It can control for dynamic factors like weather or blood pressure. And if we have more than a single subject, we can control for or estimate the effects of static factors like demographic variables. The next line is just examples of different trajectory shapes that are implemented in PersonAlytics, whether it's polynomial or piecewise or logarithmic. And then I mentioned that an important decision to be made is your error covariance structure. And we've built in a lot of automated model comparisons so that you don't have to go through the laborious process of specifying several different ARMA or ARIMA models or compound symmetry, spherical geospatial correlation structures. Those are all just output as tests, and you can use model comparison fit indices like the AKK's information criterion or Bayesian information criterion to quickly decide which error covariance structure fits your residuals. Next slide. As I was mentioning, you can use AIC or BAC to compare models. I mentioned earlier that maximum likelihood has been shown in prior studies to be your best option for model comparison. So maybe comparing a model with 2 different covariance structures or 2 different ways of structuring time. but your parameter estimates that you want to interpret at the end of your study should be estimated with restricted maximum likelihood, or REML. And then this is just a summary of what I've been talking about. The tasks that we've automated so far are residual correlation structure selection, time structure selection. We've also implemented some other tools that are nice, which include standardization of the outcomes, predictors for both. So if you want your parameter estimates on the R squared or Cohen's d scale, you can standardize those prior to running the analyses. We automate centering of the time variables so that your intercept terms are interpreted in relation to the point where you go from the first to the second phase, and automated selection of outcome distributions. As I was talking about, we can implement several different distributions, and we can get tests, BIC or AIC comparisons across several different distributions that might fit our data in a easy automated manner. Next slide. Switching gears just a little bit, building on the PersonAlytics package, which is for analysis, which is what I have been talking about. I'll talk about the PersonAlytics Power Package, which uses simulation-based power estimates. The users can input what they expect their effect sizes will be between groups and between phases, whether those effects are a jump, what we call a phase jump in a change in a behavior or outcome, or whether it's more of a linear change that can be specified within each phase. And after those estimated effect size are put in, we use simulation to estimate what power is for those conditions, and we can go through and adjust the number of participants in your study, the number of time points, the number of time points per phase, and go through several different scenarios to see what your statistical power will be. We're building a web-based GUI to implement this and make it easy for individuals to get power analysis results quick before a study. And as Ty mentioned, we recently published a paper showing results from this. Next slide. We'll show some example results from the paper, but I encourage you to go download a copy. These are power curves across different combinations of time points and different sample sizes, with the number of time points ranging 20 to 100, and the sample sizes also ranging from 20 to 100. And from left to right, these are different overall effect sizes of an intervention from a baseline phase to a post-intervention phase. And in this example, we can see if we have lots of time points and lots of individuals and a larger effect size, our power is near 0.1, but the more interesting things are on the left side of the plot, where we may be balancing the constraint of how many people can we afford, how many time points is it reasonable to get data from them, taking into account participant burden concerns, and what sorts of effect sizes will we be able to detect. Next slide. At this point, I think we're ready to start answering questions. I know I've seen a few coming through the chat, and we'll go from there.
Thank you very much for this presentation. We have lots of questions. The chat has been very active. So let's get started with these. First, I'll remind everybody that the recording and the slides will be posted on the Mind the Gap website in about 2 weeks. So don't fret if you weren't able to make notes about something. You can come back and download the slides and the recording later. So some questions. You've used, I think, R as the basis for the PersonAlytics software. Is that correct?
That is correct. We use that because it's freely available. Anyone can download it and get to use it. And it's also has a lot of flexibility in being able to implement the features I was just talking about, as well as rapidly incorporate other developments other researchers may do, because the open source platform facilitates that sort of rapid communication between researchers.
Right. One of the things that we've bumped into recently with R, which is annoying for us, and we hope that they will address it soon, is that they implement the 1997 version of Kenward Roger, but not the 2000, I think it's 2010 version, not the more recent version. Is that something that you all have considered?
That's something that we are trying to include in some of our next research, taking time to be able to add that, because that has been a problem for us. Not all of the R packages we depend on play nice with the current implementation, and it would be nice to be able to either write our own or make that more updated.
Right.
We have used also the Staterthwaite option,
right.
And the finite correction option Stephen's implemented as well.
Right. Are there you all have used R, but is it possible to look at these kinds of data, these kinds of study with other packages? I don't know what SAS or Stata or SPSS or some of the other common ones offer, or is this simply too complicated or too special for those packages to have addressed yet?
I've analyzed these data with SAS, which in part led me to the option of looking for an R option. It is a good program for analyzing these data. However, it's not designed specifically for it, and it can be rather time consuming. Getting the information from different pieces of the analysis can feel like duct taping results together that you want to get full information on. However, it can be done. And FDA, for example, requires that SAS be used for analyses. And so when I've worked with pharmaceutical companies, that needs to be done in SAS. And so we've made do with it. I've worked less with SAS and not with it. I mean, with Stata and not at all with SPSS in terms of analyzing these data. So I can't really comment to those.
No,
I can jump in and say,
go ahead.
I can jump in and say that any software that does mixed effects models can do what we're doing. We're just building a suite of tools that really builds on that. There's a question in the chat. Yes, you can use Python, mixed effects models in Python, but like Ty was just saying, we're trying to remove a lot of the tedious parts, automate them, make them easier and more accessible.
Right. So one of the things that's appealing with the software that you described is that it sounds like you've automated the process for picking a covariance structure for residuals and a time structure based on what the data have to say. When I was listening to that, I smiled and thought, well, that's going to be very helpful to people that want to know, well, how do I pick a correlation structure? How do I pick a time structure? On the other hand, traditional clinical trial statisticians would say, well, you should specify those things a priori, not pick them based on the data. What say you to that concern?
No, it's a good point. I think this falls in line with having a priori hypotheses of all kinds, as well as looking for maybe exploratory leads in your data. A challenge in this debate, in my mind at least, is that the error in the error covariance structure can lead to biased estimates, but that error could come from both directions. We could be hypothesizing it incorrectly, or our data may be giving us a biased look at what error covariance may be. So I think it's helpful to approach that from both directions to try to make sense of it. And hopefully the expertise related to a particular study will give a good lead in that direction. One example of this is that with alcohol use, for many people, their autocorrelation structure is 7 days, where they have certain patterns of use on Fridays and Saturdays, and and not the other days. And unless you go into that, it with an ARMA model that kind of fits that, you probably will not pick it up. So I don't have a set pat answer for that, but the insight is well appreciated.
We have a suggestion from the audience to use cross-validation for the selection of time and covariance structures.
Excellent. Yep.
As something that might be helpful.
Absolutely.
You know, prevention research often involves participants crossing from baseline to intervention, and they never go anywhere else. They don't go back to a control state. They don't go on to another intervention. And if that's the design of the project, the methods that you're describing here probably are not helpful. Would you agree?
I may not be understanding the question entirely, but a multiple baseline design is similar to what I think they're describing, in that in many instances, a multiple baseline design reflects the clinical experience in which someone comes in for treatment with a given ailment, and treatment is implemented. Treatments such as therapy or education can't be withdrawn, and so we're kind of stuck with that exposure going forward. On the other hand, there are times when a clinician may try a new treatment if that first one doesn't work, and that may more closely resemble a more complicated phased study. But there are so many options out there that there's probably some way to reflect a more complex intervention or simpler intervention approach that you might have in mind.
The last slide, one of the last slides that Dr. Tuller showed us in that one with the power results. The minimum number of time points is 20. The minimum number of participants is 20. You know, there might be circumstances where you have lots of time points, but only one participant, a handful. So presumably the software would accommodate that, and you just haven't shown it in this particular figure. Is that accurate?
Yes, that's correct. Stephen, it'd probably be good to comment about the software specifically, but I will mention at this point, we've analyzed biosensor data, wearable biosensors, where we were studying stress in the wearers. And the challenge there is kind of an interesting thing for a statistician, in that the glut of data is just so massive that p-values have no place anymore. It's really about, is this an effect size that's worth maintaining, and is it theoretically reasonable to keep a parameter or a variable in the model? And an example of this in our study that was actually really helpful is that we tried to control for differences between devices. Anybody who's played in this space knows that there's a lot of technology advances, a lot of work yet to do on the biosensors because of problems with the technology itself. And so we gave two different wearable devices to the same person and then controlled for those differences by dummy coding. And we're able to parse that out. And it was really intriguing because for one device, we had more of a linear sort of trend. And in the second one, we had a quadratic trend that we were then able to pull out. In addition to that, we had multiple control variables that we were able to parse out of those data before we got to our real research question. One of those was passing of time, but another one was differences between night and day. The length of time that a biosensor was being worn was important because we were looking at electrodermal activity, and sweat accumulates underneath those sensors and can accommodate electrical signals. And so we were able to also pull that out. So those are just some of the examples. Maybe Stephen, if you can comment to PersonAlytics handling of those data.
Yeah, we're working on making it as general as possible down to single subject designs. This results that we have on the screen were just the simulation study conditions we considered as a first step, but the software is flexible. I think was the question, how low can you go? You can go down to a single subject. and theoretically down to 2 time points, and then you've got a repeated measures 2 time point design. But other software exists for something that simple if you have enough individuals.
Right. I was intrigued by a comment, I think, from Ty, that ICT methods can be used to examine, and I'm paraphrasing here, can be used to examine the effects of individual components of an intervention package, or perhaps the best order for individual components within a package. Can you say a little bit more about how that's done? Because that's often of interest to people.
Yes, great question. So this would be, at least the way that I imagine this and the way that I would approach it, is to consider a component of an intervention as a phase of a study. And let's say you have a 4-phase prevention program that's delivered like a curriculum. Those components can then be ordered in different ways, and the effects then can be compared across those phases. And if you're getting an effect that's associated with, say, 1 or 2 of those components consistently, then that's the component that's probably doing the magical work that you want to see happen. So conceptually, anyway, that's how I'm thinking of it. It's not really in the analytics so much as in the way that the phases would be designed.
So that you'd set the study up so that one component is given, and then and you're collecting data for a while, and then another component is given and collecting data for a while, as opposed to all the components given at once and some analytic approach to teasing out individual effects.
That's it. Yes.
Right.
Exactly.
Right. Okay. Understood. We have heard quite a bit in the literature about using methods like multiple optimization strategy or SMART designs to look at the effects of individual components and optimizing the set that gets carried on to the full-scale trial, or to look at sequencing. How would those methods compare to what you just described with ICT methods?
Yeah, that's a great point. As I would imagine, David, I haven't thought about this in great detail yet. And I'll qualify that because we have thought about it in the context of just-in-time interventions. As I understand where these could intersect is in the frequency of data collection, so that as within an ICT context, you would keep the most or SMART designs, but be able to collect many more observations. And then if you were to reorder portions or different treatments, you'd kind of come back to what you were describing earlier about trying to identify the key components or the most effective elements of that strategy. And then carrying out those studies otherwise the same way that they're described already. And then the analytics wouldn't be that different either, as I understand it, but I have not ventured into that space myself personally, David. I'm really just speculating.
Okay. Going a completely different direction. How long do ICT trials last? Days, weeks, months, years? What is typical in your experience?
Yep, thank you. Typically what we've done, they last a few weeks is the most common time period that's been in my experience. They might go from, say, a few days up to months, is a more common range. Outside of that, you're probably on a tail end of a distribution.
Yeah, okay. So that probably means that this stands in contrast with something like a step wedge design that can go on for years. And in a step wedge design, we're very worried about the confounding effects of time. That is probably not an issue for ICT trials because they're quite short. so that it's unlikely that some external event would happen that could affect the outcomes that would need to be addressed in the evaluation of the trial.
I would say, I think that maybe the concerns about time are a little different in ICTs, because to go back to the example with the diabetes and the manual pancreas, one concern there was historical effects related to the nursing home changing its diet, or a holiday happening like Halloween or something, where it could affect the glucose levels and contaminate how we would interpret the results in that way.
Yeah, no, those are exactly the same kinds of things that could happen in a step wedge design or some other longitudinal design, but they're it's not something that's happening at a state level or a national level. It's a more local event that could affect the outcomes.
Yep, exactly.
Yep.
And so that multiple baseline is how we try to manage that. So let's say that the nursing home did change their diet and became healthier. Across all 4 participants at that date, we should see that change uniformly across them, or something similar across them. And then we can account for that change statistically using an interaction term or something to that effect. Okay. One way that we're advancing these methods is in studying communities. So instead of an individual person, we're looking at individual communities, and then using publicly available data sets like criminal justice data or emergency room data to track whether a policy or community organizing, for example, has had an impact on that community. And in that case, the timeline would probably stretch longer than what I described earlier, where it would be more like months or years. And we would need to control for things like a change in policy at a city level or state level, perhaps. But a multiple baseline design can also control for that if it's implemented the right way. I'm speaking about a specific study in Milwaukee that's ongoing that we have yet to see how it turns out, but we're trying to adapt it in that way. But it's the conceptualization and the approach and the variables that we use are quite different from a clinical study.
Okay, I'll pose one last question and then turn back to Dr. Moyer to wrap things up. In your experience and the experience of other investigators that you've spoken with, are these ICT methods well received by NIH review panels, study sections?
That's such an important question, and they have been so far. It's interesting because there's been an instance or two where I've presented these methods and someone from the audience has a knee-jerk reaction of, those are quasi-experimental designs, they're worthless. So there's thinking like that out there. But so far we've gotten lucky. When we've proposed these, the criticisms of the proposals have generally not been around the methods. And I think it's things like the power analyses and the other methods that people are beefing up and increasing the rigor around them that are very helpful in that way. It's really a match of the methods to the research question and the situation that's really important.
Well, I'm pleased to hear that study sections have been responsive to these methods when they've been presented and judged appropriate for the question that's being posed. Sometimes we hear feedback that study sections are resistant to new methods.
Yes.
Yeah.
Yes, I've heard that too.
Yeah, I want to thank you both very much for this presentation. I wish we had more time because there's lots more questions, but I want to thank you both, and I'll turn this back to Dr. Moyer.
Thank you, Dr. Murray, and thank you to everyone who participated in today's webinar. On the Mind the Gap website, prevention.nih.gov/mindthegap, you'll find several resources for this talk, including the slides and a list of references. We will also be posting a recording of today's webinar on our website in a few weeks. Thank you and have a great rest of your day.